Welcome back. So we're going to continue our discussion on biochemistry, and then we're going to look at eukaryotic chromosome organization. Pretty important concepts here, so let's get right into it. So first, we need to talk about histones. Okay, so DNA wraps around histone proteins to make what we call a nucleosome. So if we look over here, this whole thing is a nucleosome, right? So these are your histone core proteins, which are these guys, right? So you have H2A, H2B, H3, H4, and then H1 seals the DNA like we see here as the DNA leaves, okay? And them coming together makes what we have here, which is called a nucleosome. So next we need to go over heterochromatin and euchromatin. So heterochromatin is dense and transcriptionally inactive. Okay, so these two concepts are pretty important to know the difference between these two, and I look at it as like 3 points because you will get questions on it. So it's like just know euchromatin is active transcriptionally, and then heterochromatin is inactive transcriptionally, and you should be okay. And the last two, let's look at telomeres and centromeres. So telomeres are at the end of DNA strings, okay? And pretty much every time a DNA replicates, the telomere end gets shorter. Pretty much that's like a buffer, okay? So again, it's going to protect the degradation of the actual important part of the proteins. And we can tell how old DNA is actually by how long their telomeres are. So if a DNA has really short telomeres, that means it's it's an older replicated DNA versus long telomeres. And then your centromeres are going to be the middle of your chromosome. So you have like your two sister chromatids, and then in the middle would be your centromere, bringing them together. Yeah, so very simple concepts, but this is a very good foundation. If you understand these, you can tackle MCAT topics 100%. So I'll see you guys in chapter 6.3.